Hello everybody, my name is Matt. I'm one of the SI leaders for Biochemistry this summer. I teach my sessions on Tuesday, Friday, period 3. And one of the things that my students really wanted to go over in a review video was purine and pyrimidine biosynthesis. We're just getting ready to take our second exam, but we've gone through a lot of stuff with Dr. Xiley. And sometimes we just have to remember that, oh, hey, Dr. Purich did have a couple lectures at the very beginning of Exam 2 material. And unfortunately, these are some of the harder lectures from his nitrogen metabolism unit. So, don't let this fly under the radar. And this is what we're here to kind of talk about today because, synthesizing this really big molecule over here does have a lot of steps. But when you really break it down, it's not too hard to understand. And then we're going to talk about, his yellow box rule and how sometimes the yellow box rule is not always true. But, we're just going to kind of jump right into it. And I'm going to have a lot of extra pictures on the side. And I'm going to have kind of a summary list at the end of purine biosynthesis. And then once we're done going over purine biosynthesis, because this is the bigger, scarier molecule, then we'll quickly just run over pyrimidine biosynthesis. But I promise once we get done with the big one, pyrimidine biosynthesis will be very, very easy to follow. So also this program that I'm using, it does lag occasionally. So if it looks a little choppy, I apologize about that. But, the voiceover is going to be what's most important. So basically this is going to be the first slide. And you have to start thinking about, we're trying to make a purine, and it's going to be a double ring, and you have to start somewhere. What you really start with is the phospho-ribose sugar, and that's just going to be abbreviated as PRIB, and then it's going to have PP right here, and that just means there's two phosphates on the side. So basically, you have to start looking at this in segments because he says, yes, I'm only going to test you on what's in these yellow boxes. And this is where, the Purich's yellow box rule, it doesn't always apply because although these are where you're going to get your test questions, if you don't understand what's on each side and like the change that happens, then you're not going to understand what's even in the yellow box. So we're going to, just keep that in mind, and we're just going to go forward with all this. So basically, you're always going to start with your sugar backbone, basically. And the very first thing that we have to do is we go from just the pure sugar to having a nitrogen added onto it. This is just the most easy addition of a nitrogen. And you have to know that this is going to be catalyzed by glutamine addition. Now, we see this, the bucket mechanism that he said. This just means that, there's that special enzyme that has the aprotic ammonia tunnel that goes through the middle kind of right here. And, you're just going to have a very basic addition of nitrogen. So one of the themes that I'm going to show is that when you see just a very basic addition of nitrogen, you have to link that in your mind with glutamine. Because when he gives you test questions, he's most likely going to give you a before picture and an after picture, or words even, but he's probably just going to give you a structure. And you have to understand what's going on from the left and then to the right, and then you say what caused that. So right now we're talking about a basic addition of nitrogen, so or a nitrogen group, so that's just going to be glutamine addition. Whenever you see that you do add glutamine as well, you might also just keep in mind that in the reverse, you would have glutamate and two inorganic phosphates, but most of the time the forward direction is going to be what's most important. So, all right, not too bad. We got our first step out of the way. We added just a nitrogen-containing group on here. Now, step 2, it sounds like a really big process. So step 2, our starting is going to be this, and then we're going to end up with this molecule over here on the right. So step 2, it looks like a lot has happened, but if you notice, we've only added just one component, and that's going to be glycine. Now, if you look over here in what's dotted in red, you can see that this is the entire amino acid of glycine. So let me get my highlighter out. Yep, and it just started to lag again. Let me reopen this. I apologize about that. This will not take long. Here we go. Okay, so now I can get my highlighter out. So basically all that happened is we added this right here. And this is just all in itself glycine. You can see our backbone of the amino acid, or the peptide bond. You have the N-terminus up top. Let me switch back over to my pointer. We have the N-terminus up top. We have the C-terminus on the bottom. And then we have the carbon alpha in the middle, and the side chain is a hydrogen. So that's why there's just 2 hydrogens right there. So you just need to be able to see that that is glycine right there. So I'll go ahead and erase that. But you just need to be able to see that this is glycine right here. And notice that when we added a glycine over here, most of the time on the rest of our peptide backbone, there would be another oxygen coming off of the C-terminus. However, you know, this is how chemistry works. You just formed an amide bond linkage. And that's why the ATP is needed. Because when you would have, and I'll even draw this out over here, when you have the C double bond O, and then you have the O minus over here, and then you have the rest of the molecule over here. I'll just call that R. So when you have this, the whole point of adding ATP is to turn this oxygen over here into a phosphate group, OPO3. So I'm kind of drawing with my mouse right here, so I know it's not the prettiest, but the whole entire point of having ATP on here is the ATP is associated with the glycine before it gets added to the purine that you are constructing. And basically, like this R right here is the rest of glycine. So if you add the phosphate group on here, it becomes a really good leaving group. And that's what is able to form the link between this carbon and this nitrogen right here. So that's how adding glycine is achieved right here in step 2. So now, you know, so far not too bad as well. You added a whole big chunk of this ring, and that's entirely glycine right there. Notice that I do have that one initial picture all the way over here, just so you can kind of keep in mind where we're going. So over here in step 2, we got the addition of glycine added onto here. So on a test, you know, in step 1, you would see just the sugar base, and then you would see nitrogen being added. So you need to link that with glutamine. If you start with this this intermediate, where all you have is just the nitrogen on here, and then you add a whole, you have to just be able to recognize this as all glycine right here. You can see it's R chain, you can see it's C-terminus and N-terminus. If you add something really big like that, that's going to be glycine. So now we move on to step 3. And once again, you don't need to know these big names over here. He's really just going to only test you what's on in the yellow boxes. So now we still have this right here. Remember, we had this addition by glutamine, and then we had all of this being added on by glycine. So now, before we talk about what's in the yellow box, we come over here to see what happened, and we see a carbonyl group being added, and it has a hydrogen sticking off the side, and it's linked to that nitrogen of the glycine. Notice the NH2, and then now it's just NH, because one of those hydrogens, the electrons from that, a proton was lost somewhere within the organic chemistry of the mechanism, and one of those electrons is now serving as that bond formation between this carbon and the nitrogen right here. So, but we have to start thinking, okay, how do we equate this addition to formal THF? And why he put formal in blue right over here is because he's wanting you to recall a really important molecule from organic chemistry, and that is formaldehyde, which is over here. So formaldehyde, I want you guys to think of formaldehyde as it's like a bare bones carbonyl group. Because as you can see right here in the middle of this, we have a carbon and we have our oxygen. So we have a C double bond O, that's a carbonyl group. And you're never just going to have a C double bond O, just right next to each other like that in organic mechanisms, because the only way you can have C and O without any hydrogens is if you form a triple bond, and that would be carbon monoxide. So formaldehyde is, for biological reactions, a bare bones carbonyl group. So when we're adding formal THF, I want you to link that with just sticking on a C double bond O to our molecule. Now it's going to have these hydrogens that it has to carry. It's going to have this one over here and this one over here. In this case, the one on the right is serving, this hydrogen got deprotonated somewhere, and the electrons from that hydrogen, it's serving as this bond between the C and the NH, or the C and the N right here. So just notice that we added a C double bond O, and that is due to formal THF, and you need to think formaldehyde when you see this. So when you're just adding a raw carbonyl group, you are adding formal THF, and this is going to be the result. So one thing I do want to point out to you guys as well, so far this ring is not closed right now. There's a little space right in between here, so it's saying this was added with the red dotted lines around it, but this ring is not closed. So there is now it's hard for me to draw straight lines on this, but that line right there is just saying that there is no bond in between there. There's you can stick your hand through there. It is not connected at that point yet. So do keep that in mind. I'm going to erase that real quick. Okay. So seeing that this is really just formaldehyde, because he says formal transfer to an amino group. Here's your amino group over here, the NH2, and you're transferring basically formaldehyde to this nitrogen right here. That's a way to make a carbon-nitrogen bond in organic chemistry. So now that we have that out of the way, we are going to move on to the next chunk of things. Okay, so we're on step 4. So this was the end of step 3 earlier. So we have this bond, or we have this intermediate. Notice that the ring is not closed here still. So now there's one other way that we can use glutamine. In step 1, glutamine was useful. In step 1, it was only, I'm just going to scroll back up to just point at it one more time. If you're just raw adding just the nitrogen onto something, kind of out of thin air, because you have this and then you just slap a nitrogen on there, that is just glutamine. That's all you need to do it. But now we're going back to step 4, and we are now doing something slightly different, because now you see we're using glutamine and ATP. So why is the ATP there? Because we're not just adding a nitrogen out of nowhere. We're doing something a little bit more special this time. Now, in Purich's slide, his yellow box kind of cut this off, but this part of the molecule down here, it's a C double bond O on the bottom. So you need glutamine and ATP, because look at the before and look at the after right here. What's in the red dotted box is the after part, what changed. So we have a carbonyl, a C double bond O, changing to a C double bond N. Notice nothing else up top changed yet. So in order to get a C double bond N, a carbon double bonded to a nitrogen, from a C double bond O, you have to do glutamine and ATP. So you can still kind of think of it as, yes, we kind of added nitrogen in a way, but in this case we had a special modification of a carbonyl, and you have a C double bond to this N right here. Now, if you remember back to when you took organic chemistry, probably Orgo 2, whenever you have a carbon-nitrogen bond, that's really special. We deal with carbon-carbon bonds a lot in organic chemistry, and carbon-oxygen bonds, like with your carbonyls and things like that. But carbon-nitrogen is always really special. So do always keep that in mind as you're going through this. Don't forget about your Orgo. So once again, going from C double bond O to a C double bond N, that is going to require glutamine and ATP. Once again, you see the bucket mechanism. It has that enzyme, and it has the aprotic ammonia tunnel that's kind of right there in the middle. He'll put I don't think you're not really going to have to do the bucket thing on the test and really know too much about what that means. I know for my students in my SI group, I went in depth on exactly how that works. But basically, he's just putting it there to remind you that there is an enzyme that does this. And thankfully, you don't need to know the name of the enzyme because it is really, really complicated. So we went from C double bond O, C double bond N at the bottom, and we know that you need glutamine and ATP right there to do that. So now step 5, which once again, you don't need to know the names of the steps. So now the before is right here and the after is over here on the right. So what we're doing right now is, because if you think of a purine, it's a double ring structure. And step 5 is, I want you to think of it as closing the small ring. And I'm also not a fan of how he kind of rotated this molecule in this before and then the after. He rotated it because it makes a little bit sense down here in the further steps. But basically, in order to close this ring, because remember, right in between here is where the molecule is not a closed ring. There's still space in between there. So basically what happens, and I'll switch to a smaller pointer so I can kind of, not be so bulky with it. Basically what happens is you need to make another carbon-nitrogen linkage. And thankfully, whenever you had a carbonyl group in Orgo 2, we know that's a site where you can make a lot of, tertiary intermediates. If you think back to Orgo, something could attack the base of the carbonyl, and then you could push the electrons up onto the top of the oxygen, and then you would have a 4-carbon intermediate, and then you need to kick out something to be a leaving group. If you guys really hated Orgo 2, I apologize for bringing that back up, but it can kind of help explain. But anyway, so in order to close this ring, we're kind of making use of some of that. So he put these 2 little stars here because he's saying that this nitrogen right here, notice this nitrogen has 3 bonds. It has 1 bond going to the sugar base. It has 1 bond going to this carbon over here. And then it has 1 bond going to this purple hydrogen right here. So when you have nitrogen with 3 bonds, what's basically happening is you have a lone pair of electrons on the top of this nitrogen. So this lone pair of electrons is what's going to come over here, and I'm going to just draw a quick little carbonyl. This lone pair of electrons right here, this is what's going to come over here, attack the base of the carbonyl, and it's going to push these electrons up, and then you're going to have a tertiary intermediate where you have this. You'll have, now it's just a single bond because we push those electrons up, and then that would have a negative charge. And basically, the tertiary intermediate means that this nitrogen, which in this case was the nucleophile, is right here. So this carbon right here in the middle has 4 things bonded to it. So I'm not going to go through the rest of how this happens because you don't need to know it. But in order to close this ring, it is just like one of these tertiary intermediate problems you had in Orgo 2. This is the nucleophile, and this is the electrophile over here. So basically, I don't even know how it happens, but you don't need to know it either. Dr. Purich might, but that's because he's a genius. However, the nitrogen is going to be our nucleophile. It's going to attack the carbonyl base. And basically, once you kick out whatever leaving group is responsible in this mechanism, what happens is you actually have a ring closure. So, as you can see, and also the double bond will resonate over between this carbon and the nitrogen, but that's not really important. Because you don't need to memorize these structures, but you need to see before and after and make sense of what happens. So just know that, because he's only going to test you on what's in the yellow box. I probably, with all this stuff over here, I probably went way more into depth than I needed to. So you need to see that in order to get this ring closure, you need to have ATP. So think of closing this small ring on the right, you need ATP to deal with the small ring. Okay, so now we're going to go on to step 6. So now we have this closed ring, and notice, we have some double bonds that are beginning to form in the ring, and that's good. So once we have our closed ring, we need to start thinking of, or also one more thing before I go right here. Also, when you do this, the end, because this is a much more complicated reaction than even just this, what I drew over here. From this before and after, yes, you do close the ring, and also this nitrogen over here gets reduced. There's a C double bond N right here, and now it's kind of rotated on this side, but it's a C, a carbon nitrogen single bond right here. So do keep in mind that happens as well from this before to the after. But anyway, so now we're going on to step 6. We have the right side of our purine formed, and now we need to start making the bigger ring. And in my opinion, the bigger ring is actually a little bit easier, and it has some fun things that happen, in my opinion, but we'll all go over it. So right here, we are just going to, right now we're not adding a raw carbonyl group like we did with formal THF. We are actually adding a carbonyl group and a oxygen. So this is basically a carboxyl group, like C double bond to O, and then this could be O minus or technically OH. I'm going to switch over to the bigger pointer. So basically you're adding a carboxyl group in here, and really the only tool that nature has to do this with is carbon dioxide right here. I mean, you have C, O, and O. That's CO2. So you want to associate adding this carboxyl group with CO2. Because once again, you're only getting tested on what's in the yellow box, but we're going to make sense of what's in the yellow box by looking at the white. So basically, notice that this happened at the site of, this got added onto a carbon right here where my pointer is hovering over. This got added to this carbon, and this carbon has a double bond right here. So basically, we are adding something to a carbon with a double bond because it's going to be the easiest to add things to, because whenever you have a carbon that forms a double bond with something else, that means that it is missing electrons. So it's easy to take advantage and add things to a carbon that has a double bond. So basically, you add CO2, and you just have to kind of remember that ATP is required for this as well. If you just want to make the link in your head, CO2, always link that with ATP in addition. But on a multiple choice, I wouldn't see him putting just CO2 and then another answer choice of CO2 and ATP. I assume that he would give those both to you in the same answer choice if he says, what is required to get from this intermediate to this intermediate over here, from start to finish. He's probably going to put both of those in the answer choice, but just remember that CO2, you should kind of link that with ATP. So, awesome. We added CO2 onto our group, onto our purine, and you're starting to see we have the little ring on the right, and we're starting to see the big ring form on the left. So now, notice that in our final product over here, it's just a quick little reminder picture, this big ring has no oxygen within like the actual 6-membered ring over here. There is no, even in the small ring, there is no oxygen that's in the actual ring. But we kind of have one that, right over here where my cursor is now, we kind of have one that looks like it's going to be there. So that's going to be our next problem that we have to solve. How do we get that? And you can kind of look over here and cheat and see what's going to happen next. How do we get this oxygen right here to turn into a nitrogen? And that's going to be something we've actually seen before. So this is, now don't be scared by all this stuff on the screen, but this is going to be step 7. And we are going to have to run into our old friend that we saw in the urea cycle, which is all of this over here. I copied and pasted this directly from, I think it was lecture 10 or 11 or something like that, whichever one had the urea cycle. So we're going to run into our old friend succinate again. And basically succinate is a derivative of aspartate. And looking back at the urea cycle can help us see exactly, how this works. So over here, in the urea cycle, from when we went from citrulline to argininosuccinate, In this case, we had to add aspartate and ATP. Notice we are actually doing the same thing over here in our purine biosynthesis. So we're going to add aspartate and ATP. But what does this do? Because he just abbreviates this as succinate right here, suck. So we have to see what exactly is happening. So when we have citrulline going to argininosuccinate, I highlighted this C double bond O right here, and I might as well, just for to draw the parallel to it, I'm going to highlight this C double bond O, because we're working with literally the exact same mechanism. Now, I don't want you to link it with this enzyme name right here. It's definitely not argininosuccinate synthase that does this, but it has the same exact mechanistic properties as argininosuccinate synthase. So we have this C double bond O right here, or let me switch back over to this guy. Okay. Yeah, so we have our C double bond O right here, and what adding aspartate and ATP does is it makes a succinate linkage, because this right over here, in the red right here, this is aspartate right here. If you, I'll switch to black. If you kind of delete all of this stuff up here and look at only this down here, that is entirely aspartate right there. So you can tell it's aspartate because we have our C terminus right here. We have our carbon alpha right here where my circular cursor is. Here's our N terminus, which is actually linked to, in this case, citrulline. So our N terminus is right here. And remember, this is carbon alpha. So the third side to it is this hydrogen. And then the fourth side is the side chain. Aspartate has one CH2 group and then our carboxyl. So always remember your amino acids. Don't forget about these things. But basically, we're making a succinate linkage. So what this does is it gets rid of this C double bond O right here, and it makes a carbon-nitrogen link right here. And that's the nitrogen of the N terminus of aspartate, which is why we have to add aspartate plus ATP. So we have this linkage right here, N linking to succinate, is an identical linkage as citrulline was linking, or as when we took citrulline and turned it into argininosuccinate. It's the same thing as argininosuccinate linking over to aspartate right over here. So don't be scared by that. Aspartate plus ATP, if he gives you before and he gives you after, in basically test taking, like you have to start seeing this stuff. If he gives you before and he gives you after, and you see succinate is added, he might just even give you a picture of this right over here. Because technically, when aspartate is linked to something, we refer to it as succinate. He might give you all of this instead of just SUCC. But either one of those, you just need to know that that giant thing added on is aspartate, and succinate is what we call it when aspartate is linked to something. So this is step 7. We added a succinate linkage because that is somewhere within the mechanism. It takes this oxygen that's right here and it throws it out. We don't know what happens to it. We don't care what happens to it, but it is officially gone because remember, our final product over here, it has no oxygen, in the ring. And notice we're seeing the derivative of this nitrogen right here that is from aspartate. So now step 8 basically says, okay, well now we tagged this big aspartate molecule on. How do we get it off? Because we are interested in keeping this nitrogen right here. So this one right here, we're interested in keeping this nitrogen, but we don't care about the rest of succinate, or the rest of aspartate, basically. So how do we get rid of it? Basically, this is a trans elimination, and we ran into this, once again, the same exact thing in the urea cycle. And the part of the urea cycle that did this was going from argininosuccinate over to arginine. And what happens is, this is the big parent molecule, and then we have the succinate linkage down here. So basically what happens is, there's going to be a trans elimination, and we can see the trans elimination right here. Whenever you have this linkage right here, the electrons are going to want to join the parent molecule again, because we're trying to kick out succinate. Because notice in this before we have succinate, and over here we do not. So basically, the electrons from this carbon-nitrogen bond right here, I don't know if you can see that very faint pink line that's right about here, but it goes back up. The electrons in this bond, they come back up to technically form another double bond between this nitrogen and the carbon of the parent atom. However, in both of these cases, the carbon that's next to that nitrogen, because this succinate, when it kicks out, it's going to want to form a double bond between the carbon and nitrogen, but there's already a double bond up here. So basically, it's just going to be one of those electrons, and it's just going to be happy with sticking with this nitrogen, just like it does over here, because this is the result in the urea cycle. Once we kick off that succinate, the succinate is going to keep one of those electrons, and then this other electron is going to want to come back to the parent molecule, because right up here it's arginine that's going to be the parent molecule, but over here it's our purine that we're making. So this is how the nitrogen is just right here. So basically, I hope you can see the parallel between the urea cycle and purine pyrimidine biosynthesis. Once again, it's not these same enzymes that are active. He does not want us to know what enzymes do this. But you just have to see that when you add succinate onto something, you better be thinking that succinate is derived from aspartate, however you want to pronounce it. They're both correct. But you need to keep that in mind. And then step 7 and 8 really should just be one big step, because this is what's called a concerted reaction, and basically both of these steps are going to happen simultaneously, but it's just easier to see what happens when you kind of split it up into two. So succinate getting kicked out, and now nitrogen being left behind, that's just step 8 right there. That's finishing what you started in step 7. But notice going from the beginning of 7 to the end of 7, and now the beginning of 8 to the end of 8, because the beginning of 8 is the end of 7. So keep that in mind. But from the beginning of 7 to the end of 8, what happened is we literally just took this oxygen right here and we turned it into a nitrogen, and it's going to have some hydrogens just to satisfy the formal charge of nitrogen, which always wants to have 3 bonds. But look at this. We have oxygen right here, and all we did these 2 steps just to turn this oxygen into a nitrogen, basically. So, and kicking off the succinate is due to a trans elimination, and adding on the succinate is due to adding aspartate and ATP. So, and then once again, recall the urea cycle if you really want to know how this works out, because he details it really nicely in the urea cycle. So now there's 10 total steps, if I remember correctly, so we are almost done. Excuse me, that is my phone going off. I apologize about that. But basically, so we have the last 2 steps going on. So we have to now, because we got if we go kind of back up here, we got the bulk of our big ring that's forming, and now we need to finish the job. So step number 9, we are going to have to add, because if you take a look at closing this ring over here, we're not quite ready to do it yet, because if we were to close this ring as it stands right now, we'd be joining a nitrogen and a nitrogen. And the only way in nature that nitrogen is ever bonded to another nitrogen is going to be diatomic nitrogen, which is a nitrogen-nitrogen triple bond, which we know is basically impossible to separate, and the amount of energy you would need is crazy. So we are not ready to close this ring. We need to add some stuff that would be more happy to close the ring than just these 2 nitrogen groups right here. So we're going to go back to our old friend, formal THF. So once again, remember formal THF as really just adding formaldehyde. And I'll kind of just draw it again up here. Formaldehyde is a C double bond O. And then it has just the 2 hydrogens right here. because in organic molecules, you're never going to have a carbon with just a C double bond O and then nothing bound over here out here, because then it would have 2 extra pairs of electrons, and then, you know, the formal charge on carbon would be all sorts of messed up, and that's never going to happen. So remember, just formal THF, you need to link this with formaldehyde. Formaldehyde, once again, is the most basic form of a carbonyl group. That, like, if you want a raw carbonyl, you utilize formaldehyde. And in this case, it's formal THF, formal tetrahydrofuran, I believe. But once again, you don't really need to know about what that THF is. So now that we kind of revisited that, we're just going to add formal THF. And what this does is it's going to add the carbonyl onto this bottom, the purple nitrogen. You just kind of have to remember that, because technically it's a little bit easier to add it to this one because there's so much resonance going on between this carbonyl and this carbon-nitrogen bond right here. So it's going to want to preferably avoid all of this jazz going on up here because there's way too much going on. So formal THF is going to add our carbonyl, or yeah, it's going to add basically our bare carbonyl group onto the bottom nitrogen. And once again, this is another one of those things where this ring is not closed right now. There, I'm going to draw like another line just temporarily. This ring is not closed, but we have introduced a chemical group that is going to be much happier to, it's going to be much more willing to make the close, which is going to be basically step 10. So we added formal THF to add this carbonyl group to our bottom nitrogen. So now we're almost done. We're going to the last step. I'll erase that for you now. So we're going to our last step, and now, so this is the end right here, because also, once again, just always test-taking tips. You see before and you see after, we added a carbonyl, just a bare carbonyl group, and we know that that is derived from formaldehyde, so you need to think formal THF. Make that connection in your head for the test day. So now, once again, last step, we are going to want to close the ring. Now, this is a slightly different ring closure because notice there is no ATP. I will quickly scroll back up to our first ring closure of the small ring. And this required ATP. The reason why this required ATP is because we also had to deal with this carbon double bond to nitrogen down here, and basically to deal with the carbon double bond to nitrogen, you're going to have to add water, and then closing the ring subtracts water. You're going to precipitate one water molecule out. So you're adding one molecule to take care of this carbon nitrogen, and then, you know, do whatever kind of reducing, oxidizing that needs to happen there. And then you're going to technically lose a water right here, but we're not going to say water is involved with this reaction because you're adding one and then you're removing one. So the net utilization of water is 0. So I want you to just in your head think that when we close the small ring over here on the right, I just want you to associate that you need ATP to do that. But when we close the big ring, so scrolling back down here now, when we close the big ring, we don't have to really deal with any carbon double bond to nitrogen anywhere, because, you know, there's no carbon double bond to nitrogen anywhere present here. So we don't need to add a water just to get rid of a water. We're only just going to lose a water. So in this case, once again, dealing with the carbon double bond to nitrogen is why we needed ATP. So there's no ATP. However, we also have to, you know, close this ring, and we're going to lose one molecule of water in the process because we're really only looking at the forward direction. If you want to memorize the reverse direction for all of these, like if you have time to do it, go ahead. But you can see how the reverse direction is just the converse of the forward direction. Formal THF going forward up on the top, and then THF going backwards on the right, or on the bottom. You can recall that if you want, but he's mostly going to talk about the forward reaction, I promise. So, basically to close this ring, how are we losing water? Well, one of the hydrogen, because we're losing H2O, so we're losing 2 hydrogens and we're losing 1 oxygen. So the first hydrogen comes from one of these hydrogens over here on the NH2. So that's going to be one of them. And then we're losing this hydrogen on this carbonyl, which was derived from formaldehyde. So this is one of the hydrogens, and this is another one of the hydrogens. And then the oxygen that we lose is this, it's from the C double bond O. We are losing this right here, because technically this is another one of those tertiary intermediate reactions. I drew it above a little bit earlier in the video. You can look at, just the basic behavior of a tertiary intermediate. We'll just go quickly back up and show it to you one more time, just to drive home all of that. Because in the bottom, it is a nitrogen attacking the carbonyl base, and then you have a tertiary intermediate, and when you're done kicking it out, you have just the ring closure in this case. So going back down to it, because this nitrogen over here on the left is acting as the nucleophile, because it has 1 bond going from the nitrogen to the carbon, and then 2 other bonds. So you know that nitrogen has a formal charge of 5, which means there's 1 lone pair of electrons, so there's 2 little dots up here, and that serves as the thing that basically attacks the base of this carbonyl and then makes the tertiary intermediate in order to do the ring closing. But I hope that by bringing this up, your Orgo 2 bells in your head kind of go off a little bit. But basically what ends up happening is you close the ring, and then there is a double bond formed right here. And basically, in most tertiary intermediate reactions, instead of having a double bond, there would normally be another molecule that could donate some electrons so that a double bond wouldn't need to form. However, in this case, there's so much resonance in the molecule that it's actually favorable to do this, because you complete resonance, because the last part of this resonance could be from the C double bond O resonating down to being right here if it really wants to. So there is full resonance that's achieved, and that's the last step of making your purine. So basically, one thing you need to know is that the result of all 10 of these steps is the molecule IMP. And IMP it's an intermediate that it looks like adenine and it looks like guanine, which are 2 nucleotides in our ATP, in our DNA. So basically, you make IMP because it's a nice molecule that can easily form AMP and GMP, which is just the base A in our DNA and the base G in our DNA. So these things out to the right, these are just from my little personal notes. These are no longer notes that are part of the video over here. But basically, you're at IMP, and he goes over a little bit of the regulation of this. I'm not going to dive too much into this because this is a little bit easier to understand. Basically, in order to form AMP, your body, it's only going to do this when there's plenty of G. Now, this is faded out right here because he explains it later. But when you have plenty of G in your body, if your DNA has plenty of G to work with, it's going to say, hey, let's just make some A instead because I have plenty of G. Because A is base paired with T, G is base paired with C. So in this case, it's just going to take IMP, and it's going to use GTP, because making the base of A in DNA uses GTP. The base A in your DNA is very closely related to ATP, and the base G in your DNA is very closely related to GTP. So when guanine is very present within your body's going to be like, okay, let's make some ATP. So it's going to use the energy molecule for G, which is GTP, and it's going to call upon our old friend aspartate again. And it's actually going to do, you don't have to know as much chemistry on this, but basically, look at before. We have a C double bond O. look at after. We just changed this oxygen to a nitrogen. Think of changing an oxygen to a nitrogen, especially a double bonded oxygen to a single bonded nitrogen. You're going to have to go through this whole succinate thing. Adenylosuccinate. I don't even know how to pronounce that, but we see succinate again, and that's what this whole entire thing tagged on up here is. So you're going to use GTP as the energy source, and then aspartate, and then this is going to tag on succinate. And once you do another, up in our pyrimidine biosynthesis that we did above, we said that there's a trans-elimination that kicks this off. And fumarate is actually, that's what we call succinate, which is a derivative from aspartate. That's what we call it when it gets kicked off. And what stays behind is this nitrogen. This aspartate molecule up here, which we call it succinate when it's a linkage, it donated its N-terminus to the parent molecule that's down here. So we use, when we have IMP and we want to make the base A, we use GTP as the energy source. We go through our old friend aspartate to make a succinate linkage, which donates its N to the parent molecule, and now we finally have AMP. Now notice there's stars next to these, so you do have to know these structures. That really sucks. There's nothing I can do about it, but that's part of why this course is really hard. Even right here, he put this in its own little box. GTP provides energy for AMP synthesis, because when you're trying to make AMP, your body is telling you that you have plenty of G and GMP available. So it's going to drive it the other way. So this is the mechanism that he kind of went over. You don't have to know any of this. I promise you will not get test questions on this. So I'm just going to do a big X over this in the video, just so you remember that, because these mechanisms are way harder than we can even do in this course. So, and they're even simplified in that picture I just X'd out. So just really quick, going from IMP to GMP, because once again, IMP, it's ambidextrous. It can do both of these directions. Basically, what we are going to do right here is look at the very end product. The very end product, we're not caring about this C double bond O up here. We like that in this case. We don't want to add succinate to turn that into a nitrogen in the GMP pathway. We're going to deal with this region right here, and the end goal that we want to do is put a nitrogen basically sticking off of there, because right here, this is all from the start to the end, we're just adding a nitrogen basically with its hydrogens to complete the formal charge. So what we have to do is we have to add we're going in this direction. Okay. So what we have to do is, so the bottom, we're going to introduce NAD+, and NAD+, what basically happens with this is that NAD+ is lacking electrons, and NAD+ will very strongly take electrons away from something. Excuse me. So we are going to add NAD+ to get rid of this double bond. Literally, NAD+, it just comes along, and it's going to take away some electrons. And now, whenever we start with a double bond, that already means that we don't have enough electrons. Like, theoretically, we do not have enough electrons to complete everything, like, perfectly. That's what double bonds are for. It's made to compensate for a loss of electrons. So, in one step, we are adding NAD+ and H2O. So, NAD+ will take away even more electrons, but that'll make it a prime time for water to just come over here and form a double bond with its oxygen. Notice the 2 electrons in water, because if you have a double bond right here, that means you're missing 1 electron somewhere, so you'd make a double bond to compensate. And now NAD+ removed another electron. So now we're missing 2. Well, remember that hydrogen is the vehicle for electron transfer. So how convenient is it that water has 2 hydrogens, aka it has 2 electrons. So it kind of bonds onto here. It takes care of the missing of 2 electrons by this double bond in NAD+. So now its oxygen just makes a double bond right here to the carbon, and now all of the formal charges are basically taken care of. This intermediate is called xanthocene 5' phosphate. You don't really need to know this intermediate. There's no star next to it, thankfully. And then lastly, we're starting with a C double bond O, and this is a little bit different than up here, because over here we went from a C double bond O and in the upper mechanism, and then we ended up with just a nitrogen. However, what's interesting to note is that we are not going to use the succinate thing again, and that's because there is no double bond at the base between the carbon and the nitrogen. In this case, no, in this case, it's just, I misspoke on that. Don't listen to that, what I just said right there. But in this case, the final product that we're going to want to make, it's a different enzyme, basically. So kind of forget what I just said a second ago, but in order to turn this oxygen into a nitrogen and form a double bond right here, we're going to have to use ATP and glutamine. Yeah, so that's how we're going to do it, and GMP is the final product. Notice we're using ATP to make GMP, and in the above, we were using GTP to make AMP over here. Okay, so that's everything you need to know about making purines. So what's awesome is that purines, it's a much bigger molecule and it's much harder to do. So pyrimidines are going to be a lot easier because it's just one smaller ring. So we'll just quickly scroll down to here. Once again, this is the mechanism of going from IMP to AMP and stuff like that. You don't need to know any of this. That is the really harder mechanism part. He said to mark that as reference and this as reference as well. So we're getting that out of here. And this, even though this is my notes and I made this really big, this is the regulation slide. Read over this. It's really not too bad once you figure out, it's a pretty well-made slide, and it just says, at certain parts of this, your body will say, hey, I have enough GTP, go over and make AMP. I have enough AMP, go over to make GMP, whatever, you know what I mean? And it's saying it can be regulated at any of these steps. Most of the time in fall and spring, Dr. Xyley will have taught all of his material before we ever see nitrogen metabolism. And then regulation in the fall and the spring, it makes much more sense, but it's a little bit different because we did nitrogen metabolism before Xyley did his stuff. Okay, so now we have, so that's just the regulation stuff. Once again, you can go through that. I'm going to just scroll on down to our pyrimidine biosynthesis. I need to close this out again because it's lagging. I took that picture in the background, by the way. Fun fact. Amateur photographer, I guess. So we're going to reopen this real quick. Here we are. Okay. Oops. So we're going to get into our pyrimidine biosynthesis, which is going to be nowhere as bad. Oh, also while I'm here with you guys, I had a direct test question on this, what I circled right here, or let me get this pointer thing out again. All right, I apologize. This is lagging once more. I have to get rid of it. All right, hold on. It's booting up. Here we go. Oh, OneNote. Okay, so basically, I just wanted to quickly, while we're here, I had a direct test question on this. Gout is treated with literally this drug right here, allopurinol. It was a really easy question, but this is just a quick reminder that literally almost everything is exam eligible. Gout is treated with allopurinol. That was a test question for me from Purich, and it could be one for you. Okay, so basically we're going to go over the last little bit, pyrimidine biosynthesis. It's going to be really easy because the purine was really, really hard. Now, I don't have this recurring picture next to everything, but we can see that so far, aspartate is really responsible for almost everything. So we're just going to jump right into it. So making a pyrimidine is a little bit easier. We're going to run into our old friend CPS2, and we need to, our first component of making this pyrimidine is carbamyl phosphate. Excuse me. And carbamyl phosphate is made by using glutamine and ATP, and it has, we've seen this before, CPS2. That's how you make carbamyl phosphate, and it has the little bucket mechanism again. So now back into all this stuff. If you take carbamyl phosphate, the whole point of making carbamyl phosphate is to put a phosphate group onto a carboxyl, that's the whole reason that you do it. So you put this phosphate group on here because that serves as an amazing leaving group. So if we take, in step 2, carbamyl phosphate and we add this molecule right here, it has some enzyme. I don't think you need to know that enzyme. It wouldn't hurt to, but I don't think you need to know it. This is aspartate. We have our C terminus right here. We have our N terminus right here. You have the hydrogen sticking off, and then all of this on the top is the R chain up here. And we can see that aspartate, once again, it has one carbon and then the carboxyl group out here. So we know that's aspartate. So carbamyl phosphate and aspartate, you can see how this leaving group of phosphate group is being kicked out. This is doing another tertiary intermediate thing. This nitrogen is attacking the base of the carbonyl, which pushes the electrons out up here on top of the oxygen, which then, once the leaving group is kicked out down here, then one of the lone pairs of electrons slams back down to make this double bond, and then that's how you have the linkage between all this. So the after, this is the before, and this is the after. If you see the before, which is literally just this little guy right here, carbamyl phosphate, and you add this giant thing onto the side, you need to just be able to pick out that, hey, we have a C terminus right here. We have an N terminus right here. That would make, this is the C terminus, this is the N terminus. That would make this thing right here in the middle the carbon alpha, and then you see a side chain, and that's the side chain of aspartate. So this process is called carbamylation, but you're just doing the tertiary intermediate trick because the phosphate is a really good leaving group, and now you have almost all of the ring right here. So that's step 2. You add aspartate, and we're almost there already. So now we're going to go on to step 3. These notes on the side are, once again, my own personal notes from when I was re-listening to the lectures with you guys. So over here, we're going to have the before picture, because it's what was left over from step 2. And now we are actually closing a ring. And are we dealing with a carbon double bond to a nitrogen anywhere? No, we are not. So you don't need ATP to close this ring. But we are going to lose water. We're going to lose one of the hydrogens from this. We're going to lose one of the hydrogens from this. And then the oxygen that gets lost is, in this case, the oxygen that gets lost is also this oxygen right here. Because this carbonyl stays, because that's right there. This carbonyl down here stays. That's over here. So, we lose this hydrogen, and then we lose this oxygen. So that's where the minus H2O happens in this step. So once again, no problem. We closed a ring that does not involve C double bond N, carbon double bond nitrogen. So we don't need ATP. We're working with losing H2O. That's all that's happening in order to close this ring. It's a dehydration. And now going on to step 4, we have our closed ring, and this is a little bit tricky. You have to look at the before and you have to look at the after in this case. The before, nothing on the left side really changes. That carbonyl is still here. This nitrogen is still here. This carbonyl in red is still there. This nitrogen down here is the bottom. It's still there. This carbon, this carboxyl group down here, it's still there. But what happens, I know I went through everything, but what happens is, this is why he wrote all these hydrogens sticking off, because he wants you to realize right here that these 2 hydrogens, basically they're gone. We lost these 2 hydrogens, and whenever you lose those hydrogens, you also lose an electron, because once again, hydrogens are the vehicle for electron travel. So we're losing those hydrogens, and in order to make up for that, these carbons, because each one of them lost one hydrogen, so they lost one electron. So they would normally have like a little dot next to them, like they were a radical, but obviously we don't like radicals in organic chemistry. So we are just, I'll erase those now. So instead of doing, like having two radicals right next to each other, that's exactly why double bonds form, so that we don't have to deal with radicals. So you use NAD+. Once again, NAD+, it really wants electrons. So it's going to rip the electrons off of here. It's going to rip those two off and become NADH. So, and when it does that, it's going to leave behind a double bond. So technically, we eliminated both of those hydrogens, but NAD+ is going to come around and it's going to oxidize this because the molecule lost electrons. If you remember oil rig, oxidation is losing electrons. So our parent molecule got oxidized. Therefore, NAD+ got reduced because NAD+ gained the electrons. Reduction is gaining. That's where the rig comes into play. So now we have this, and it's starting to look really close. So we're going to oxidize to form a double bond. If you want to read those notes on the side, but those are my own personal ones. So now we go on to step 5. There's only 6 steps in this instead of 10. But these steps are much easier to follow because we went through all the hard stuff with purine biosynthesis. So we are going to, right in here, notice we have the ring that's been forming, but we don't have the sugar base. In purine biosynthesis, as opposed to what we're doing right now, pyrimidine biosynthesis, in purine biosynthesis, we had to you had to deal with the ring before you dealt with the sugar base. But when you're making pyrimidine, it's a much smaller molecule, so you don't need to worry about adding the sugar until close to the end. In the bigger molecule, like a purine, it's more advantageous to get the sugar associated with the base of your ring first, because it's such a really big molecule. And if you were to try to add the sugar to the double ring that's already formed, there'd be so much steric hindrance going on that like it would be hard to do it at the end. So that's why you start with the P-ribose, basically, the sugar at the bottom. In purine synthesis, you do that first because it would be way too hard to add it at the end once you have two rings formed above it. But with a single ring, like in a pyrimidine, it's no big deal. So in this case, if you look at the before and you look at the after, you notice that nothing in the ring changed except for you made a linkage of the PRIB, the phosphoribose sugar. Like, I will kick your guys' butts if you do not say that on a multiple choice. If he says, like, you know, what catalyzes, or not what catalyzes, but how do you get from point A to point B? If you don't pick like this as the answer choice, I'm going to go over there and kick your butt because this is the easiest one. You just literally add the sugar base on right here. Notice what's written below here is in gray. You don't need to know that because there is some really fancy mechanism as to how this happens, but it's way beyond us in this course, and we don't really care about it. And it's awesome to say we don't care about some of this stuff because it's really hard. But anyway, so we are almost done. We added our sugar base on, and now we have to take care of the last step. And this is really going to bring back the Orgo 2 for you guys. So basically, if we look at our before and we look at our after, what's missing is actually this entire carboxyl group. And how do we just get rid of it? Because the carboxyl group is nowhere to be found right here. How does this even work? So basically, what you have to, because obviously you can see this arrow right here, we are losing a CO2. What you have to remember from Orgo is that what's happening right here is a decarboxylation. And in organic chemistry, in the organic chemistry lab, you can do this with just heat. However, in biology, we have enzymes that do things much faster than you can do in an organic lab. Enzymes are super catalysts, basically. So here's, to bring back some Orgo knowledge from you, we have a carbonyl group right here. Yeah, we know that's a carbonyl group. So if we go over one carbon, we call this the alpha carbon. It's a little bit different than carbon alpha. That's what I use to talk about the peptide backbone of the amino acid. But we're saying this is an alpha carbon right here. So if you have a carbonyl, and here's the alpha carbon, and here's the beta carbon, the next one further away from it, decarboxylation can happen when another carbonyl group is beta to the carbonyl group of reference. This is your carbonyl group of reference up here at the top. I'll circle that. This is your carbonyl group of reference. This is alpha. I cannot really draw with my mouse on this to make these letters. And this is beta. Right here. This is the beta carbon right here. And when you have, because this is a C double bond O, it is a carboxyl group. When you have a carboxyl group that is beta to a carbonyl, a carboxyl group that is beta to a carbonyl. I think before I said a carbonyl that's beta to another carbonyl. No, it's a carboxyl group that is beta to the carbonyl group of reference. This is eligible to decarboxylate. So now that we talked about that, I'm going to erase this just to make it cleaner again, and that's how you do it. OMP, I would definitely know the name of this enzyme because it's in the yellow box. OMP decarboxylase, because this is OMP. Really, you just need to recognize that it has decarboxylase on there, because most of the time enzymes are named in a way that helps you remember what it does. So it's taking OMP and it's doing a decarboxylation. So in the organic lab, you could heat this molecule up and make it decarboxylate on its own, but that would require heating our body temperature up way higher than, 98.6 degrees Fahrenheit, whatever that is in Celsius, you know what I mean? The normal body temperature. So this enzyme is going to finish our pyrimidine biosynthesis. So we are done with pyrimidine biosynthesis. I know that was a lot. This recording so far is 1 hour and 7 minutes. So feel free. I mean, if you made it to this far and you didn't speed it up already, like you could have done it from the start. I know that I like to go really in depth onto things. And some of you guys might already know some parts of this because you've already studied or it makes more sense to you. But I had a lot of my students say, hey, this stuff is making my head spin. It's really complicated. But basically, we are pretty much done. It's all just a matter of looking at the before and looking at the after and getting comfortable. And this is where studying comes in. I would recommend you can make flashcards of a before and an after, and then you have to tell yourself what goes in the blank. You put the answer on the back of the flashcard, or do whatever you want to do. So, this is pyrimidines down here. But up here, I'll scroll back up to, once again, because I kind of, for purine biosynthesis, which is much harder because, once again, it's a double bond, or it's a double ring. For purine biosynthesis, on the last slide, or on the last thing that I kind of talked about it with, I made a little list over here on the right side, which we're almost back up to it. I'm going to have to close this out because it's lagging again. I'll pull it up. We get to look at this nice picture in the background. How awesome. But let's see. Let it load back up again. Yeah, so we're just going to keep on, because on the very another way to, you can make like flashcards having a before and an after picture, and then you fill in what goes there. However, for the purine biosynthesis, I put this little thing up here. How far up is this? My goodness. Okay, so I put all these words over here to the right. And for purine, I did this for purine biosynthesis, and I'll let you do it on your own for pyrimidine biosynthesis if you really want to do it this way. Because, you could do, or let me pull out my pointer notation thing again. Because you could do a before and an after flashcard, and then you write in what goes in here. You could also choose to study it this way if you want. What I basically did for the purines is I put over here in bold all of the molecules that you use. So, kind of just scrolling up, I got this all from here. So I took this picture apart and I said, okay, how do we assign what all of these things do? Because it has arrows pointing at what all these molecules do. But now, going back down here, I put this all kind of into words. So basically, if you want to add a nitrogen, in this case, we're going to use glutamine. That was our first step. You added just a raw nitrogen onto the phospho sugar base. So that was just glutamine. Now, in order to basically, because we go, if we're talking about glutamine plus ATP, remember that involved a carbon double bond to an oxygen, and then, because we're still technically adding nitrogen out of nowhere, but we're adding it to a C double bond O and changing it to a C double bond N. In both these two steps right here, we are adding nitrogen out of nowhere, but in order to add it to a double bond, you're also going to need ATP to make that happen. This one's kind of simple. When we added glycine as one of our steps, you just have to be able to point out that glycine was added because it looks a little bit bigger. You'll see the N terminus, you'll see the C terminus, you'll see the carbon alpha that has 2 hydrogens on it. So when you add what looks like glycine, you just have to be able to pick it out. That's a little bit harder of a question he could ask you. But, just glycine is there. So adding a raw carbonyl group uses formal THF, and we talked about it up here. This is formaldehyde. Formal from formal THF comes from formaldehyde. So that's how you add just a raw carbonyl group to something. If you see carbon, if you basically see you're adding CO2, and in this case I should have said adding a carboxyl group. I can actually change that right here. So instead of CO2, we could say adding carboxyl. Adding carboxyl group, that is going to be done by, and I'll switch back over to the second point. Adding a carboxyl group is achieved by using CO2. When you close the small ring, note that ATP is used. Final big ring, I'm skipping down a little bit, is a dehydration. It's just minus H2O. So in the small ring, there's no H2O for closing the ring. Well, technically there is, but you use H2O and then you lose an H2O. So the net H2O involvement, it's 0. It's like a spectator ion. It doesn't formally participate because you add it just to take it away later. So it's as if it wasn't there anyway. So think in the purines, when you have the double ring, you close the small ring with ATP. Changing a C single bonded to an oxygen to a carbon single bonded to a nitrogen. I kind of worded this differently, but we're talking about the aspartate through the succinate linkage trick, and we've seen that many times in this video. So, I'm just, instead of doing a before and after picture and looking at it visually, I'm putting it into words right here if you want to study it this way in addition as well. But you're probably going to be given pictures. But you can use this as a key to kind of see it in a different way. And then getting rid of succinate, just remember that was a trans-elimination. We can kind of see it a little bit up here. We got rid of this succinate up here in the corner by the trans-elimination, and that's how you kick off fumarate. And remember the urea cycle. Once again, the urea cycle was really instrumental in learning how this works. That's the first time we ever encountered it. But, so yeah, that's basically everything I wanted to go over with purine and pyrimidine biosynthesis. Hour, 15-minute long video, but speed it up a little bit if you want to. It would probably take you a couple hours to study this on its own anyway, so maybe this video is a good pace. I don't know. I really like to go into things in depth with my students. But yeah, so we're basically going to end it there. Feel free to share. I'm sharing this with my SI students right now. However, I've kind of been talking as if I'm talking to other students as well. So feel free to share this video around. I'm going to upload it to YouTube later. And I hope you guys do well on the test. This is, in my opinion, purine and pyrimidine biosynthesis is the hardest single concept to learn in this course because there's just so much going on. There's so many big molecules being drawn, and you have to know what's in the yellow box, but you also have to know what's on the outside of the yellow box so that the inside of the yellow box makes sense. AKA Purich's yellow box rule isn't 100% true. I made a meme about that in my SI group because, we like to have fun in our group as well. But, the studying, I can't explain this enough. I was in your guys' shoes. I took this class not too long ago. And, although I'm an SI leader, so I got an A in the class, I had to work my butt off to do it. So that's why I like to put a lot of effort into videos like this to kind of really explain in a way that I think you guys can understand. And, I'll throw in some Orgo 2 knowledge so that you feel as if you are, you have useful knowledge that can help you figure this out. But, I just like to do that, and I like to be as clear as I possibly can. So I wish you guys the best of luck. And don't forget, study as much as you can for these tests because sadly only the top 15% of the class gets an A. So just worry about beating the average and, you will fall into your category of whatever your grade is based off of there. So wish you guys all the best. Have a good day.